Okay, well, thank you. And I click on, I got it. Okay, I'm Ed Levin, Director of the Duke Center on Addiction and Behavior Change, part of DIBS, and welcome to our seminar series. This is our penultimate talk for the fall of 2021. We do have one more next week. Connor O'Neill will be speaking to us about evidence-based treatment for co-occurring substance use in adolescents and young adults. He's actually from Duke, from the psychiatry department. And then before I introduce today's speaker, I just wanted to kind of look ahead. So after next week, we'll be taking the break and people can have a happy holiday season and continue to work in their laboratories and clinics and do all that good stuff. But we're going to reconvene on January 19th with Linda Perino from Wake Forest University, who's long been a leader in the field of drug abuse research. And she's going to talk to us about consequences of chronic drug use and reflections on a career in translational neuroscience. Then we're going to have later in January some participants in the TROSA program. That's a local program in Durham to treat the substance abuse, and it's a tremendously successful residential program. And then we're going to continue on through the semester. But I did want to highlight the symposium we're going to have on February 22nd, when Together for Resilient Youth, Dr. Wanda Boone and her colleagues there in Durham on marijuana legalization, brain science, community impact, and risk prevention. So all of this information is available on our website, our webpage of the DIBS site, and then also I think Tyler was going to list it in the chat as well. And then I am starting to gather information for the fall of 22, believe it or not. We're not even in 22 yet, but starting to work on that. So if anyone wants to give a talk or suggest a speaker, please let me know. And so now I'd like to introduce today's speaker. It's our very own Dr. Andrew Hawkey from the Department of Psychiatry and Behavioral Sciences here at Duke University. And Dr. Hawkey earned his bachelor's degree at Elon University. Actually grew up here in the Triangle area, and then went for a master's at University of North Carolina, Wilmington, and earned his PhD at the University of Kentucky, and has been here at Duke for the last 4 years or so. And he's going to be talking to us today about smoking risks in pregnancy beyond nicotine. Andrew?
All right. Well, thank you all for being here today. Today, we're going to be talking about smoking in pregnancy. So we know there are lots of reasons to stop smoking and to encourage people to stop smoking. But one of the issues that we sometimes forget about is pregnant women and their ability and the support that they need to stop smoking during their pregnancy once they find out that they're pregnant. And much of the focus in this area has been on nicotine, but it's important to know that there are thousands of chemicals in tobacco smoke. So what I would want to do today is to talk about one area of this that many of you will be less familiar with, which is looking at combustion products, particularly polycyclic aromatic hydrocarbons. So for today, I'm going to be presenting just behavioral and developmental data from our research group. We do have some excellent colleagues who've been doing work in this same area, looking at neurochemical effects and metabolic effects of PAH toxicity. But we'll be concentrating on the behavior today. So let's start by orienting ourselves to where we are when it comes to smoking. So as we're all well aware, overall smoking rates have been in decline over the last few decades. And even so, in the US, it's still estimated that around 1 in 5 adults use cigarettes on a regular basis. But we are seeing dramatic declines in the use in adolescents and in young adults in particular. At the same time, we also have additional routes of smoking that are increasingly popular in our country. So with that, we're talking about marijuana, which despite having THC and cannabinoids as their primary constituents of interest, this smoke also contains many other compounds that are of interest for tobacco, as well as other forms of toxicant exposure. And overall, the number of people who have been who engage in marijuana use, particularly young adults, has been relatively stable as rates of cigarette smoking have been in decline. And there are some hints of increasing use in recent years, which we will likely see continue to change as legalization continues. I do want to make one important note about pregnant women, though. So tobacco products are still among the most used drugs of abuse during pregnancy. It's estimated that upwards of 10% of pregnant women report cigarette use during their pregnancy at some point. And marijuana use is less common, but still a major concern. So 5% or potentially more of pregnant women report the use of marijuana during their pregnancy. And this is a major concern for a variety of reasons. In part because women don't find out that they're pregnant immediately. And so problematic use of most drugs of abuse is most pronounced during the first trimester. But when you look at cigarettes in particular, we actually have very low rates of quitting success among pregnant women. So it's estimated that only around 40% of pregnant women who smoke successfully quit at some point during their pregnancy. And there are certain high-risk populations that have even lower rates of success, even down to, say, 20%. So this is an area that really needs some attention, and in particular, trying to find the best routes for encouraging successful abstinence during pregnancy. So since smoking drugs of abuse is still a major issue of concern, I do want to give some attention to the route of exposure. So smoking has some very unique properties when compared to things like vaping and smokeless tobacco. Principally, one of the differences is the presence of combustion products. So one major class of combustion products is polycyclic aromatic hydrocarbons. So these are products of incomplete combustion that can take a wide variety of forms. Chemically, they have a similar multi-ring structure, but they can come in many shapes and sizes. And through their interactions with our developing bodies, they have many different potential impacts on the way that we develop and the physiology of our cells. And ultimately, when you look across different adverse outcomes, you see that PAHs are relevant to a wide variety of potential outcomes, not only neurotoxicity and teratogenicity, but also things like endocrine disruption and reproductive disruption. Also issues like oxidative stress and genotoxicity and immunotoxicity. And they're perhaps best known as carcinogens. So what we're dealing with here is a relatively unique concern that's going to be really relevant when we're talking about smoking during pregnancy. So I would like to make a quick note, though, that cigarettes and marijuana are not unique as routes of exposure for us with PAHs. So when you look across our society, PAHs are actually a major concern for environmental health broadly. So things like wildfires and coal production and coal ash management are major sources of air pollution and water pollution when it comes to PAHs, as well as the use of fossil fuels and a variety of industrial products like creosote. So all of these can contribute to secondary exposures through air, water, and soil, and a variety of products that we may come into contact with. For those of you who like smoked meats, there's also substantial concern about adding PAHs to our diet through those particular routes. So if you're someone who really likes to grill, this isn't exactly great news. Now, in terms of how these operate, PAHs work very differently than most drugs of abuse that we're interested in. So they don't act primarily through a membrane-bound receptor. Instead, they are associated with the Ahr genomic pathway. So Ahr is the aryl hydrocarbon receptor, and this is a ligand-activated transcription factor. So essentially what you have here, you have this transcription factor that works with coactivators to manage the expression of our DNA, the transcription and then expression of our DNA. And we have endogenous ligands that work through Ahr, but those can be mimicked by many of these exogenous ligands like PAHs. And because this is a transcription factor, the effects that it has on those cells, particularly in development, can be really, really widespread. So when you look at cellular development, really all levels of development have vulnerabilities that are related to these genes and this altered transcription. Everything from proliferation to differentiation, and then on to circuit development. So when we look at the developing brain, we do see that Ahr is a heavy influence on development in many areas of the brain, particularly the cortex and limbic system and the hindbrain. And so with this, there are quite a few vulnerabilities that we need to be concerned about for how Ahr agonists may influence cellular development and ultimately tissue construction during critical phases of development. So what does this ultimately mean for brain function? We have these toxins that have a myriad of different effects based on at least one central mechanism. And ultimately, the nervous system is particularly vulnerable to these things. So everything from the blood-brain barrier and the integrity of the BBB is vulnerable to PAH effects, and Ahr is a major mediator of the development of that integrity. Additionally, neuroendocrine systems, such as sex hormone pathways and thyroid and pituitary hormones, all rely on Ahr as an underlying mechanism. And then with respect to neurotransmission, many of our main actors of interest have Ahr as an underlying component. And critical proteins are going to be influenced by Ahr agonism. And then looking at non-neuronal cells, so microglia, oligodendrocytes, some of these other supporting cells can also be disrupted by Ahr agonism. So this is a very complex problem, and there are many layers to it. But ultimately, it goes back to preventable exposures to PAHs and other Ahr agonists. So as I said, there are many potential outcomes, but I do want to give you one classical example that can show us how these toxic exposures may translate to neurologically relevant behavioral effects. So one of the well-developed adverse outcome pathways for PAHs involves glutamate. And in particular, changes in the expression of critical subunits within NMDA receptors that are critical for long-term potentiation. So one of the things that we see when we are looking at, let me get a laser pointer here. One of the critical things we see that's relevant to Ahr is a modulation of the expression of the NR2B subunit that is a component within the NMDA receptor. And we see that there is a dose-dependent reduction in the expression of that subunit within areas like the prefrontal cortex and the hippocampus that are really important for higher cognition and learning and memory. When we look at the electrophysiology of those circuits, we can also see changes in their adaptive processes like long-term potentiation. So if an electrical stimulus is provided within the hippocampus, typically we'll see an increase in a long-term increase in firing within that population of cells. But exposure to a classic PAH like benzoate pyrene can lead to blunted long-term potentiation, which could potentially help to explain why there are connections between learning disabilities and exposure to PAH-containing mixtures. So what we've got here is something that can help to show how these molecular events ultimately lead to longer term and persistent changes in the function of our brains. But ultimately, that connection is one that needs substantial testing and substantial validation. So within the human literature, there is some suggestive data suggesting that PAH exposure during pregnancy in particular and early in life can lead to anatomical changes in brain structure that are correlated with changes in things like processing speed and other basic cognitive processes that can be tested in young children. And using standardized batteries like the BSID, we can also see developmental delays emerging, where individuals with higher levels of exposure to PAHs early in life by 3 years of age are showing impairments in neurotypical function at that particular age. And these are things that emerge over time. So what we're really looking for here is, again, a good justification for connecting these particular problems with the PAH exposures themselves. So let's get down to brass tacks here. How confident are we, given what we know, that PAHs are actually the cause of developmental problems that are associated with smoke exposures? Ultimately, human studies have provided us with some really useful associations between PAH-containing mixtures and a variety of developmental outcomes. Everything from birth weight and head circumference, so physical development, to developmental delays, hitting developmental milestones at appropriate times, and a variety of psychiatric outcomes, particularly in childhood. So issues like ADHD, autism, mood disorders, and learning disabilities all have established associations with early exposure to PAH-containing mixtures. But ultimately, these mixtures do provide us with some issues in trying to show that connection in a really valid and compelling way. So the data that we have from humans does have some really important caveats that we should touch on. The most obvious of these is that PAH exposure is unevenly distributed across the population. It is a central issue within environmental justice. And so there are many subpopulations in the US that have other conflated risk factors for developmental issues that are also at higher risk and have higher rates of smoking and secondary exposure to PAHs through other sources. So what we need to then do is try to understand those conflations and try to parse out what the actual effects of the PAHs are in those associations. The secondary problem is that even though we look at PAHs and their toxicity on an individual basis, these chemicals are not presented one at a time. They are virtually always present as part of a mixture containing a variety of related and unrelated chemicals. So when you're dealing with a plant-based smoke, you have a variety of alkaloids coming from those plants. You can have heavy metals. You can have a variety of other established teratogens that are part of that mixture that again muddy the water when we're looking at PAHs and their particular risk factors for development. So these are serious caveats, and they're not issues that we can easily dismiss. So we do need other forms of data that will provide a more compelling link between PAHs and long-term changes in development and cognitive function. So how are we actually going to do that in a precise way? Ultimately, in the Levin Lab and in the Superfund Research Center, we use animal models principally as our risk assessment tools. And we can look at the effects that PAHs have on development in multiple ways, including structural and functional components. So we can look at primary birth defects using models like Zebrafish that are excellent for that type of work. And we can look at long-term changes in more subtle aspects of how tissues develop and ultimately function. And then additionally, we want to show that those changes structurally have a functional relevance. So they are connected to psychological outcomes of interest for children and developing humans. So I'm going to start the talk looking at data with something that's probably a bit unfamiliar to most of you, and that's going to be looking at zebrafish. So zebrafish are a really interesting model that allow us to look at development in a really comprehensive way while using a much simpler model organism with which to test. So our model for zebrafish exposures, looking at pregnancy equivalent issues, is to provide embryonic exposure from gastrulation to the inflation of the swim bladder. So this is a period of time that essentially covers the development of the animal from a very early stage in cellular development before we have separation of the early cells into layers to the point where the fish are hatched and freely swimming. And so covering that period, we can then look at how those exposures are going to lead to short-term and long-term effects on behavioral functions and provide a proof of concept for PAH toxicity and its relevance to behavioral functions coming from nervous system damage. And ultimately, one of the things we can do with these studies is perform longitudinal analyses and look to see how the stage of development influences the expression of behavioral problems. So the first analysis that I want to provide for you for PAHs is going to focus on benzoate pyrene. So benzoate pyrene is one of our gold standard, well-studied and well-established PAHs in neurotoxicology. And so the way that we'll look at this in zebrafish is by trying to establish behavioral tests that have a translational relevance to things that we would study in rodents. So one of our primary tests would be using something called the novel tank dive test. So this is a zebrafish equivalent of an open field test that you would use in rodents, with the critical difference being that zebrafish live in a three-dimensional environment and explore not only side to side, but also up and down. And zebrafish do show thigmotaxis towards the bottom of the tank when placed in a new environment for the first time. So with this, we can look at locomotor activity, but also that anxiety like diving that matches the thigmotaxis we see in rodents. Now, one of the interesting things that we saw with BAP is that we have a distinct pattern of hyperactivity among young animals. So 2.5 months would be equivalent to an adolescent zebrafish. And with this, we saw a non-monotonic increase in activity in these young fish. So this was our lower dose that produced the hyperactivity and not the higher dose. Interestingly enough, as these animals matured into young adulthood and then later adulthood, we saw that those effects early in life did attenuate. So this actually fits nicely with some of our understanding of ADHD, where the hyperactivity symptoms that you see in ADHD individuals are most pronounced in younger individuals. And as maturation occurs, hyperactivity symptoms are shown at a much lower rate. And adult ADHD is not as closely associated with hyperactivity as it is with attentional issues. So this does give us some lens into the developmental timing of some of the issues that we're going to see. And it shows us that the effects that you see in maturation may not be representative of the entire developmental trajectory. A second analysis that we did at multiple time points was looking at startle. So with this, we have zebrafish in these circular wells, and we'll deliver a startling acoustic stimulus by firing a pin into the bottom of each cup. And then that will happen 10 times in 10 minutes so that we can look at changes in their response to that particular stimulus. Now, unlike in rodents, a startle is represented by an increase in activity as opposed to freezing. So what we do is we will measure the amount of activity 5 seconds before the tap as our pre-tap activity and 5 seconds after the tap as our post-tap activity. And so this gives us an idea of the magnitude of the startle response. So BAP dose-dependently suppressed pre-tap activity when we were looking at young adults. And so this represents a reduction in baseline activity that was unique to this higher dose BAP exposure. But there was no change in post-tap activity. As these animals aged, however, those patterns shifted. So when we looked at the older adults, we saw no change in baseline activity, but we did see increased post-tap activity, so an enhanced startle response. And this was something that emerged as the animals continued developing after that so-called mature stage. So this shows us that not only can behavioral effects attenuate as we mature, they can also emerge as we continue to develop. So this again suggests that longitudinal studies are very informative when looking at the expression of these particular psychological impairments. So one of the central questions that we've had recently with respect to BAP effects is whether they are ultimately preventable. So we do understand the mechanisms related to PAH toxicity to a reasonable degree. And so those mechanisms present some targets for intervention. And one of the ones that we wanted to look at through a study that was headed up by Zaid Holloway, one of our recent postdocs, was looking at vitamin E supplementation as a reducer of oxidative stress. And so here we're looking at tocophersolan, which is a water-soluble version of vitamin E, and looking at that as a rescue agent that could prevent larval toxicity in our zebrafish model. So what we're looking at here is a traditional larval motility test. So we're going to shift the animals from lit to dark conditions multiple times. So under light conditions, the animals have relatively low levels of activity, and under dark conditions, they have higher levels of activity. So they are inhibited in the dark and stimulated in the dark and inhibited in the light. So with our target dose of 5 micromolar BAP, we saw reduced activity in the dark phase. So this makes the levels of activity more comparable between lit and dark. That's reduced discrimination between light and dark conditions. With the 1 micromolar toco, we did see a reversal of that particular pattern, suggesting that suppressing the oxidative stress during exposure may actually have some protective effects. Now, I do say that with one caveat, which is that the dose of our vitamin E is really important to whether you're going to get that protective effect or not. So when we looked at lower doses and higher doses of TOCO, we saw that there was no compelling reversal of the effects that we see with BAP. And so when we combine that together, we see that overall we do have evidence for an intervention effect, whereby the overall levels of activity for animals exposed to 5 micromolar BAP were improved when moderate and higher levels of TOCO were being simultaneously exposed. So there was some evidence for protection. However, this was only strong enough to reach statistical significance with our moderate dose. So there may be some potential benefit from looking at avenues like vitamin E supplementation, but across the dose ranges that we may want to look at, there does appear to be an ideal range that too little or too much may negate those effects to a certain degree. So with what we've talked about so far, we've seen some evidence suggesting that BAP is neurotoxic using the Zebrafish model, and that oxidative stress may be an important component of that toxicity. But I mentioned earlier that PAHs are found in mixtures that may contain a variety of PAHs and a variety of unrelated compounds, including heavy metals. So when you look at analyses of cigarette smoke, you do see a wide range of potentially toxic heavy metals. And so one of the things that we've tried to do recently, and this is an ongoing project, is to look at interactions between benzoate pyrene and cadmium. So the central issue that we're trying to look at with these mixtures is whether the effect of BAP is predictable when BAP is being presented alone or as part of a mixture. So previously I showed you some data suggesting that BAP at higher levels of exposure at 5 micromolar can reduce larval motility, and particularly in the dark. Now, with the 3 micromolar, we did find a main effect of BAP that was not altered by the co-presence of cadmium at 0.1 micromolar or 0.3 micromolar. But this is larval motility looking at the distance moved. So this is our sort of raw score of locomotor activity. We can also pull out other measures that may be informative in other ways, including the amount of time that the fish spend mobile. So with this, we actually found quite a different story. So BAP in and of itself is again associated with a reduction in activity in the light or in the dark, and this is being measured as mobile time over each 10-minute phase. But when BAP is co-present with cadmium, it does not appear to induce that effect. So any differences that you see based on BAP appear to be unique to single exposures to BAP with respect to motility in the dark. When you look at motility in the light, you actually see the opposite pattern, where BAP would not generally affect this outcome, but in the presence of cadmium, it does lead to a reduction in motility in a phase of the session where you wouldn't normally expect to see it. So what we're seeing here is fairly strong evidence of an interaction where the effects that you would predict based on a BAP exposure differ depending on other co-exposures that may be likely based on the mixture they're a part of. Now, I did say that we don't necessarily expect these effects to be stable across development. And so one of the additions to this particular study was looking at juvenile fish. These are 1-month-old fish. So they're not yet adolescents, but they're no longer larvae. And at this time point, we actually see a very different pattern. We don't see a pronounced interaction when looking at distance moved or mobile time. So the effect of BAP is negligible at this time point, but cadmium in and of itself has a meaningful, leads to a meaningful reduction in motility that we did not predict based on the larval fish. So again, these longitudinal studies are going to help us flesh out when and under what conditions these behavioral changes will be expressed. As we move on to adolescence, this was our first opportunity to look at novel tank. So again, the overall activity and the depth that the fish were swimming in a novel tank environment. And we had previously shown that BAP is associated with hyperactivity in adolescence. But interestingly, when BAP was present with cadmium, we did not see those effects. So, and cadmium did not have any effect in and of itself. So again, the effects that are associated with BAP may be relatively specific to BAP and not be well predicted within mixtures. We were also able to do the TAP test with our adolescent fish, and overall there was a main effect of BAP where the startle magnitudes increased based on BAP exposure with or without the mixture. And we did see an interaction in addition to that, that was time sensitive. So when comparing BAP alone to our control, we saw that these fish had higher startles towards the end of the session than you would expect. And so this is related to an impairment in the reduction of the startle response over multiple repetitions. Looking at the mixtures, we saw a relatively similar pattern for the low dose mixture. And for the high dose mixture, we actually found a more quadratic function as opposed to a linear function. And so we see elevated startle early in the session, early to mid in the session, that disappeared as time went on. So we are seeing some evidence for mixture effects that are distinct from the effects of individual exposures. So the key question when we are working with zebrafish is how predictive these observations are going to be when we're thinking about mammals. Because zebrafish are really valuable for a variety of reasons, many of them practical, but ultimately there are major differences in neurological structure between fish and mammals. So we've also performed some studies looking at rats and essentially tried to see how predictive and translational these zebrafish studies are, and how well we're going to be able to take those observations forward into the clinical realm ultimately. So, so the study that I'm going to be talking about today with rats is looking at osmotic pump exposures throughout pregnancy to nicotine, benzoate pyrene, or the combination of the two. So for those of you unfamiliar with osmotic pumps, so these are implants that can go under the skin that have an inner reservoir where we'll put the contaminant of interest, nicotine, BAP, nicotine or BAP, and each rat would then have 2 of these implants under the skin on their backs that would provide single dose exposure or the multiple compound exposures for the mixtures. So these implants are going to slowly release the compound for up to 4 weeks and cover the entire length of pregnancy and into the nursing period following that. So one of the things that we'd like to see if zebrafish are going to translate well into rodent models is evidence for hyperactivity across development. And with this study, we saw that nicotine in and of itself did not increase the levels of activity for either male or female rats. BAP, however, was associated with hyperactivity, both in adolescence and in adulthood. But it did so in a sex-specific manner. So typically we see a pattern where females are more active in the figure-8 maze than males are. And exposure to BAP reversed this, negated that sex difference. So it brought males up to female levels of activity in the figure-8 maze. So what we're seeing here is a sex-dependent effect of BAP on activity. And it also suggests that the effects that we associate with tobacco containing both of these compounds may be more associated with PAHs than it is with nicotine directly. And importantly, this was consistent across our multiple phases of testing in adolescence and then into adulthood. For a more direct set of observations with respect to sex differences, let's also take a look at fear-like responses. So with the novel environment suppressed feeding test, we are taking rats and putting them in a place they've never been in before that is bright and bare. So they're in a normal rodent cage with nothing on the floor and no lid on it. So the animals are very exposed and will have a prominent stress response. And one of the things we know about stress is that it suppresses appetite. And so you will get a lengthy period of time where the rats refuse to eat, even if they have been food deprived since the day before. So we've taken these rats with 24 hours of food restriction and then provided them the opportunity to eat in a stressful environment. And we typically see that males have wait longer to eat than females do when presented with this scenario. And interestingly, nicotine and BAP both suppress the sex difference. So you'll see equivalent levels for males and females for both nicotine and BAP. Interestingly enough, the combination of the two did not accentuate this effect. So we did not see additivity when these two exposures occurred at the same time. So they were essentially redundant with one another. Now, most interesting to me, we also looked at attentional function. And it had previously been shown that males exposed to nicotine during pregnancy show attentional deficits. And we did see that looking at our main attention task, looking at their ability to select the correct lever based on the presentation of a light. So this was a sustained attention version of the signal detection test. So males exposed to nicotine do have impaired accuracy when a cue has been presented, but co-exposure with BAP actually reversed this particular deficit. And I'd also like to emphasize that in this data set, we did not have accuracy differences between males and females among controls. But when BAP was present, either alone or in the mixture, you're seeing separation between the males and females. So we are seeing examples here where PAHs could disrupt a typical sex difference or lead to the generation of novel sex differences that you wouldn't normally see. So this tells us quite a bit about how mammals react to PAHs. So mammals have an endocrine system, and in particular, a sex determination program that much better resembles ours than zebrafish. So determination of male versus female in fish is very complicated. It's not based on the XX, XY chromosome structure. But overall, our rodent data is suggesting that sex differences are going to be critical components of the behavioral effects that you'll see based on these early life exposures during pregnancy. I will just quickly mention that we do have some ongoing experiments extending this work, looking at BAP and cadmium. So this will provide us another avenue for looking at comparisons between zebrafish and rats and show the consistency of our conclusions across model systems that are both vertebrates. Now, coming back to smoking for a minute. So there's a lot of discussion about how best to address smoking during pregnancy. And one of the things that has been presented as a possible route for improving cessation is encouraging the use of non-smoking based products. So the use of vaping, nicotine patches, various other forms of nicotine exposure. And so the toxicology of these two things will be quite different based on the presence or absence of PAHs. That much is clear. What is less clear is whether that is advantageous for all systems that you'd want to look at. So we did find evidence in our nicotine BAP studies that removal of PAHs may improve some outcomes, like the hyperactivity, but may have no effect, as we saw with PAHs in novel environment suppressed feeding, where nicotine and BAP were redundant with one another. So removing PAHs may not improve those types of outcomes. And then with attention, we saw that BAP actually reversed the effects of nicotine. And so removing that may accentuate certain problems. So there's still a lot of questions left to answer about this. But I think what we can say is that we need to directly answer that and not simply assume that because it's a simpler mixture, that it is inherently safer. So just to make a few closing remarks, animal studies do strongly suggest that PAHs are neurotoxic and can lead to behaviorally and cognitively relevant changes across the lifespan. But our longitudinal studies suggest that even though we often talk about these effects as being persistent, development is actually very critical in the expression of those particular problems. And so we may see life stage dependent changes in the particular phenotypes that we're examining with our animal models, and that may lead to differences in humans as well. And so in the ways that we evaluate these problems in the clinic, we may need to take that into account. And further, to take into account sex differences that do seem to be a major modulator of vulnerability. Additionally, animal models overall do effectively demonstrate these risks in a controlled setting, and they allow us to more compellingly draw a link between PAHs and developmental dysfunction that gets around those caveats that we had about human testing. And it also helps us to elaborate on the relative risks for different compounds within these mixtures and distinct characteristics of the mixtures themselves. I do thank you for your attention. I'd like to say a quick thanks to our Duke contributors in the Duke Superfund Research Center, and including the triumvirate of Ed, Ted, and Fred, and Rich DiGiulio, who worked with us on the zebrafish work. So I did not present any of the neurochemistry or metabolic data, but we have a really great reservoir of knowledge at this point on some of the long-term effects on the brain as well as behavior. Additionally, we've had some excellent trainees working in the lab over the last few years, including our postdoc, Zaid Holloway, a master's student, Jonah Boyda, and our excellent technicians and students. I'd also like to give a quick thanks to the financial support that we've gotten from NIEHS and EPA through the Superfund Research Center and the Niches program. Thank you.
Well, thanks, Andrew. That was a marvelous set of studies. Of course, we collaborated on these, so it was all quite familiar. But it's I think important to know for the group as a whole and beyond here that smoking tobacco is not just nicotine effects. So and you've cited the benzoate pyrene as the kind of a prototypic polycyclic aromatic hydrocarbon, but there are others, and also these metals and 4,000 other compounds. So in terms of other potential bad actors, I mean, do you think that the other PAHs are going to act pretty similar to BAP or not, or just a matter of studying them, or what?
Yeah, so they should have many of the same underlying characteristics, but they're also going to have differences in potency. And some of Rich's work has suggested that the co-presence of different PAHs can affect absorption of any particular PAH. And so in some cases, you would expect multiple PAHs to have additive effects. But if that's the case, if you're going through the placenta, going through the blood-brain barrier, something along those lines, you may have some pharmacokinetic differences that lead that to be equally unpredictable.
Great. Are there other questions? I have more. I have never-ending supply of questions here. I was wondering, in terms of the age effect functions. We've talked about this numerous times, but I think it'd bear having a discussion here in terms of the predictability of later behavioral effects from what you might detect early on in terms of larval testing in the fish and early behavioral testing in the rats. I mean, what's your take on? Can you predict what's going to happen later on, or is that pretty much a different story?
So at this point, it's not very clear, primarily because when we see a shift from age to age, we're still not really sure why there's a shift from age to age. So for the locomotor data for the fish, for instance, we could see that overall activity does increase from adolescence to adulthood, and then is relatively stable after that. It does go a little bit up towards later adulthood. So we can start to hypothesize about that, but it's not really clear which developmental processes are being accelerated in an example like that. It does appear that the adolescents with BAP exposure are showing levels of activity that are more comparable to adults than they are to adolescents. So it's possible that trajectory is accelerated somewhat, despite the fact that body size is still much smaller than it would be in adulthood. But some of those shifts in arousal may be occurring earlier than they should. On the flip side, you may also have, it may be unrelated to that portion of the trajectory. It may be a distinct neurotransmitter difference early that is attenuated through neuroplasticity. So I think ultimately we'll need to add some mechanistic work for that to really pick one option or the other.
Okay. Again, open to any other questions? If not, I have more questions.
Ed, this is Jeff. I just have one, and again, this is a state question about human expression. With tobacco smoke and pregnancy dramatically going down over the last 70 plus years, and I'm thinking particularly those of us who are baby boom generation types who were where smoking was pretty common to present, where smoking is getting fortunately less and less. I don't seem clinically to be seeing a reduction in attentionally challenged patients of mine. In fact, if anything, I'm seeing that increase. And I just wondered thoughts about that.
Yeah. So one of the peculiar things with attention and toxicity is that So attention and hyperactivity are endpoints for a wide variety of seemingly unrelated toxins. So everything from alcohol to tobacco to lead to insecticides. And so ultimately we're being exposed to a variety of these over this period, and we're aware of some of them, like alcohol and tobacco, and many of them we're not. And when you look on the industrial chemical side, we're increasingly seeing a broader diversity of chemicals being used, seeing them in household dust, seeing them in air pollution, seeing them in our water. And as there are more and more, we seem to know less and less about which risks are being driven by which exposures. So a great example is something like PFAS that's being increasingly found in water sources all over the US. And that is, so we've got smoking decreasing, and then we've got things like PFAS that are increasing. And so, the trajectories for that over time may be based on some of those alternating trajectories, where some things are going down and some things are going up. The other thing we'll have to consider with PAH is marijuana, and increasing access across much of the U.S. And potentially increased frequency of use, even if the number of people willing to use it is staying relatively stable. So ultimately we may get some replacement of tobacco with some of these other alternatives, like marijuana, like vaping, things along those lines.
Other questions? I just wanted to maybe touch just briefly on the comparison of the fish and the zebrafish and the rats. Do you think that we have a pretty broad database with nicotine, benzoate pyrene, other substances in zebrafish and rats? Are zebrafish a good model system for mammalian neurotoxicity?
In some ways. So I definitely think as a first option, when we have many compounds to screen, they're a really great model. So with a complex mixture, you have many options to work through and many mixtures to work through. And it's not terribly feasible to do all of that in rodents. So we have to be very selective in what we work through in rodents, but we can use the practical advantages of the zebrafish to work through a larger array of potential outcomes, and then prioritize what we're then going to take forward. But I will say, because of the reduced complexity of the zebrafish, it's still very difficult, as you know, Ed, to make a one-to-one connection. What the zebrafish can show very conclusively is a proof of concept, that you can have, if you're rating potential bad actors on toxicity, we can look at the potency differences. We can look at the target systems for which compounds cause scoliosis and which cause heart defects, and which are primarily neurological, and start to lay some of that out in a way that it fits the ecology of the fish and the organization of their system, even if it may not be a one-to-one of hyperactivity to hyperactivity, fear to fear. Ultimately, a zebrafish doesn't have a hippocampus, doesn't have a prefrontal cortex. So there are limitations, but as a check yes or no, is this toxic? And then rating high to low, is it very toxic, moderate, or low toxic? You can lay out quite a bit of that.
Good. Okay. Well, I wanted to thank you again for giving a great talk and a very broad view of this developmental neurotoxicity from nicotine and benzoate pyrene. And I want to thank you for informing us about that. Just wanted to remind people we're going to meet again for the last seminar of the fall semester. Connor O'Neill from Duke is going to talk to us next Wednesday, December 8th, on evidence-based treatment for co-occurring substance use in adolescents and young adults. Okay, thank you. Have a good day.